And lastly, I also want to introduce myself and my co-host. My name is Phi Wen, and I'm a grad student in the Myelination lab at UCSF. And my co-host is Ilya Weinstein, who's a postdoctoral fellow also at the Myelination lab at UCSF. So without further ado, I'd like to introduce our first speaker, Dr. Ashley Nemes-Baran. So Ashley completed her PhD in biomedical sciences, focusing in neuroscience at Kent State University and the Epilepsy Center at Cleveland Clinic. She then did a postdoc at the University of Pennsylvania and at the Cleveland Clinic Lerner Research Institute in the lab of Dr. Tara De Silva, where she conducted the work she'll be presenting today. Fun fact about Ashley, she loves cars and has built and raced several cars throughout her life. So it definitely sounds like we have a lot to learn from her. And before I hand it off to Ashley, if anyone has questions during the talks, please ask and type them into the chat box, and we'll get to them at the end. So it's all yours, Ashley.
Thank you so much, and this is a great topic. I can't wait to hear from all of the other speakers. I'm thrilled to share our work from the De Silva Lab with you today. This was recently published in Cell Reports. Our paper was titled Fractalkine-Dependent Microglial Pruning of Viable Oligodendrocyte Progenitor Cells Regulates Myelination. Here's an overview of the findings of this work and what I'll be talking about today. First, we observed that amoeboid microglia are invading the corpus callosum to engulf oligodendrocyte progenitor cells, or OPCs, during development. And the majority of these engulfed OPCs are viable or alive. And we also found that fractalkine receptor deficient microglia exhibit a reduction in this engulfment process, and fractalkine receptor deficient mice have reduced myelin thickness later in adulthood. So I don't have to talk about this too much with this crowd, but here's a simple schematic of development of microglia. They are myeloid progenitor cells derived from the yolk sac, and they infiltrate the central nervous system during early embryonic development. But there's a population of amoeboid microglia that enters the corpus callosum through the lateral ventricles postnatally, and this is the population that we're interested in this paper. They do eventually mature and become ramified pretty quickly in development. And these are just some coronal cut sections from CX3CR1 GFP reporter mice showing this population, and as you can see, they kind of congregate around the lateral ventricles and move into the corpus callosum, and they kind of fill in by postnatal day 7 throughout the corpus of genu. And if we look at P7 more closely and we evaluate the cortical microglia compared to this corpus callosum population that I'll be talking about, the cortical microglia are more ramified in their morphology. They have these long processes in a much more smaller cell body compared to the amoeboid shape of the corpus callosum microglia that we're interested in. And studies have shown that the nervous system undergoes a remodeling process throughout development. And this has been shown that it's an activity-dependent manner to make the brain more efficient for communicating. And it's known now that this is due to microglial pruning of synapses, or synaptic pruning. And recent work has shown 3 different major mechanisms that might be involved in this process. In the hippocampus, barrel cortex, and olfactory bulb, fractalkine signaling has been demonstrated. And if you look at a CX3CR1 knockout mouse in the hippocampus, we can see an increase in dendritic spines. And similar results have been found with another mechanism, triggering receptor expressed on myeloid cells 2, or TREM2, also in the hippocampus. And with TREM2 knockout mice, there's another finding of increased dendritic spines in the hippocampus. And then a third mechanism that's been demonstrated is the complement system, specifically in the visual system. And by knocking out complement receptor 3, there's a reduction in synaptic pruning and a reduction in synaptic particles found in microglial cells. And similarly, there's also neuronal pruning that's going on to fix the nervous system during development. And this also happens in adulthood. So this is an image of a dentate gyrus, or the adult dentate gyrus, with an immature neuron being engulfed by a microglial cell. And this is during neurogenesis in the adult. But it's also observed in the developing cerebral cortex with immature neural precursor cells being engulfed by microglia, as well as in the retina with retinal ganglion cells being engulfed. And if you look at development in the mouse, this makes a lot of sense. Microglia are invading early in development, and there's neurogenesis and synaptogenesis occurring just after that. And we know that microglia are involved in pruning synapses as well as neurons. But I just mentioned there's this amoeboid population of microglia that are entering the corpus callosum postnatally. Interestingly, this is coincident with the peak time point in oligodendrogenesis. So this led to our first research question: are OPCs also pruned during development, similar to neurons and synapses? To study this, we used coronal brain sections and looked at different time points in development. We identified microglia using, in this case, IBA1 with green and OPCs with reporter mice. And we took Z-stacks from different parts of the corpus callosum. We ended up with 9 per animal. And by taking those Z-stacks, we could use software to 3D reconstruct them and look at the interactions between microglia and OPCs. And we noticed in these images that OPCs were being contacted as well as engulfed, so they weren't necessarily being engulfed in all instances, and we wanted to study this further. So we quantified this engulfment using Imaris software, and to do this, we created a surface render on top of microglia, as you'll see in this video in just a second, in green, shown here, and then another surface render on the OPC shown in red. And then using Imaris, we were able to identify areas on the OPCs that were internalized within microglia, shown in gray on these images. And we could calculate the percent of internalization by cell, taking this gray surface volume and dividing it by the total surface volume for that cell. So then we could parse out cells that were just being contacted from those that were being engulfed. And we found that analyzing this data across different developmental time points, peak engulfment was occurring around postnatal day 7. And it was significantly reduced by postnatal day 11. Now, looking at some representative images across development, we see that the amoeboid microglial population was most amoeboid around P7, and they continuously ramified throughout development. However, the microglial contacting of OPCs continued to increase throughout development. We wanted to know if these OPCs that were being engulfed were actually being phagocytosed in the lysosome. So we took slices from reporter mice expressing GFP on microglia, and OPCs were expressing TD tomato. You can see some examples from these representative images of OPCs being engulfed by these arrows. And we stained this tissue for CD68, which is a marker of the lysosome. And looking at these merges, we can see there is a large part of the microglial volume that's taken up by that OPC, or by that lysosomal marker at P7, but it's also got a lot of OPCs within that microglial compartment. And then we looked at the OPCs that were being engulfed by microglia and found that the majority of them were inside of the CD68 positive lysosome, concluding that OPCs are being phagocytosed in the lysosome. Now I have a video for you to look at some interactions in live imaging. So we used confocal time-lapse microscopy to evaluate this using our reporter mice, and you'll see microglia in green and our OPCs in red. And in some situations, as you'll see by this arrow, microglia are contacting OPCs, and then they move on and go on to engulf a different cell. Then in other situations, as you'll see by this arrowhead here, microglia contact and immediately engulf the OPCs. So I'm going to start over. There we go. So this OPC will be immediately engulfed by this microglia cell, whereas this OPC gets contacted, and then the microglia moves on to engulf a different cell. Looking at this zoomed in, we can kind of clear it up or make it a little bit more clear. This microglial cell will eventually engulf this OPC, and we found that once that process is initiated, it takes about 20 minutes to complete. And since we looked at our live imaging videos and we found that microglia were engulfing some cells, and in other cases they would contact an OPC and not necessarily engulf it, we wanted to know if these cells were alive or dead, because we know that microglia commonly engulf dead or stressed cells. So we next tested the research question, are OPCs dead or alive when they're engulfed? And to do this, we looked at paraformaldehyde fixed frames first. And again, from our reporter mice, we identified cells that were contacted and engulfed, and they were mostly negative for caspase-3, which is a marker of apoptosis, and that's quantified down here across 6 different cells, or 6 different mice. And then we also looked at phosphatidylserine, and this is a marker of cell stress, and we saw the same thing. So again, you can see examples of OPCs that are being engulfed as well as contacted, and they're negative for phosphatidylserine, and this is quantified down here. But then we also wanted to look in our live imaging setup. So we showed that the majority of our cells were negative for caspase-3 as well as phosphatidylserine in fixed tissue. And we took our live sections and we used this PSVU dye. And what PSVU labels fluorescently is negatively charged phospholipids like phosphatidylserine. These are commonly expressed by cells as a target to be engulfed or phagocytosed. They're commonly called eat me signals. And we found most of our cells that were being engulfed were negative for PSVU. We did see some debris, as you can see by this asterisk, that were being engulfed by microglia, but they were negative for our OPC dye. So to look at this even further, we've shown that the OPCs that are being engulfed are negative for cell stress markers as well as cell death markers, but we haven't really evaluated their viability. So we took our live imaging setup and used a 2-dye system. we used oxazole blue, which is a marker of dead cells, simultaneously with DIL-C15. And this is a fluorescent dye that's expressed in the presence of mitochondrial membrane potential, and it's commonly used as a cell viability dye. By using the dyes simultaneously, we could look at cells that were being engulfed and see if they were expressing a marker of cell death or cell viability before being engulfed. And we quantified this and found that the majority of cells, again, were not expressing oxazole blue when they were engulfed or contacted, so they weren't dead, but more interestingly, they were expressing this DIL-C15 marker, indicating that they were viable and alive. And this was pretty interesting and exciting and also surprising. We normally think of microglia clearing dead cells and debris, but microglia can engulf viable cells. And this has been termed in the literature as primary phagocytosis or phagocytosis. And it's been more recently observed in the nervous system with non-apoptotic neurons being engulfed by microglia in situations with ischemia, retinal degeneration, as well as epilepsy. But more interestingly, during development, neurons have also been observed being engulfed alive within the cerebral cortex as well as in the retina. So now we found that microglia are engulfing viable OPCs. It's happening in the white matter at a very specific time point during development, but we wanted to know at what stage in the maturation or in OPC maturation are they being engulfed. So we took paraformaldehyde fixed sections and we stained for different markers across the oligodendrocyte lineage at P7. So this is the least mature and this is the most mature. And we found that the majority of cells that were being engulfed were expressing NG2, and they were within this pre-myelinating phase of maturation. And importantly, we did not observe any engulfment of MBP positive cells. And since the cells are in this pre-myelinating phase when they're being engulfed, we wanted to know, does impairing microglial engulfment of OPCs impact myelination later in life? But to answer this question, we would first need to block this engulfment process. And fractalkine receptor is expressed on microglia in the central nervous system, and the knockout mice are widely used tools for studying synaptic engulfment in postnatal life or early postnatal development. But also, the ligand for fractalkine receptor, CX3CL1 or fractalkine, is expressed on OPCs, but then downregulated in mature oligodendrocytes. So this made it a very attractive candidate for us to block this engulfment process. So we used CX3CR1 knockout mice compared to controls to look at this. And we found that the knockout mice had a significant reduction in this engulfment process compared to controls, as shown by these representative images in our quantification, but there was no impact on their ability to contact OPCs. There was also no change in the number of microglia or their volume. And as I showed you earlier, the OPCs are being engulfed while they're still viable. And this is a classic example of primary phagocytosis or phagocytosis defined by the elimination of viable cells. And an important feature of phagocytosis is that blocking this process prevents the loss of the target cells. So then our next question was, does blocking microglial engulfment of OPCs result in more oligodendrocytes later in life? So we took the knockout mice shown on the bottom here, compared to their controls at postnatal day 30 and stained for OLIG2, which is a marker of oligodendrocytes across the lineage, so all oligodendrocytes regardless of their maturation. And then we also stained for CC1, which is a marker of only mature oligodendrocytes. We found that the knockout mice had an increased number of oligodendrocytes overall, but also they were most of them were expressing CC1, indicating that we didn't have an impact on their ability to mature. And since we saw that there was an increased number of oligodendrocytes at postnatal day 30, we also wanted to know, does this impact myelination? So we looked at the tissue with electron microscopy to evaluate myelin thickness. And we could, as you can see in these representative images, the knockout mice did appear to have thinner myelin than the controls, but we wanted to quantify this. So we used G-ratio analysis, which is where you take the diameter of the axon and divide it by the axon with the myelin together. And a higher G-ratio indicates thinner myelin. And that's exactly what we saw in our knockout mice. They had significantly higher G-ratios, which was also significant across axon diameter and plotted by relative cumulative frequency. But there was no difference in the number of axons compared to controls or the percentage of unmyelinated or myelinated axons, indicating that we didn't change anything with the axons. We just basically found that the knockout had thinner myelin. So to conclude, our results showed that the amoeboid microglia that are migrating from the ventricular zone into the corpus callosum at early postnatal development are engulfing OPCs alive. And this is termed in the literature as primary phagocytosis or phagocytosis. And microglial engulfment of OPCs is a fractalkine-dependent mechanism, and blocking this process results in an increased number of oligodendrocytes, and that leads to a hypomyelination phenotype. So this suggests that this is a really important process for normal white matter development. We're really excited to continue exploring this further. And I'd like to acknowledge the people who helped with this project. Obviously, my mentor, Dr. Tara De Silva, and our co-author, Donovan White, as well as the rest of the lab. We also got a lot of help from the Cleveland Clinic Imaging Corps and the Cleveland Clinic Art and Photography Department. And we also want to acknowledge our funding sources. And thank you for your attention, and I'm happy to take any questions.
All right, that was an awesome talk. If anyone has any questions, please type them into the chat box. As questions are rolling in, I'll start off with a question of my own. So for the OPCs that are getting eliminated, you've shown that they don't seem to be undergoing cell death. So I was wondering, what your thoughts were on what makes the engulfed OPCs different from the spared OPCs, and if there's any sort of, functional heterogeneity in these OPCs.
Yeah, that's a golden question that we're exploring next. We're trying to now look at, we've blocked a process through fractalkine signaling in the microglial cell. So now we want to turn to the OPCs and see what they're expressing as their ligand to be basically being tagged as an eat me signal. We've looked at phosphatidylserine, which is a common eat me signal for stress cells, and I showed that these cells are alive and viable. But yeah, the next question is what could be that ligand on the OPC that's being targeted for engulfment? We know that these cells are pre-myelinating. We know that they're alive, but why would they be engulfed as opposed to other cells? So that's the next question our lab is hopefully going to get at next and try to turn to the oligodendrocyte to figure that out.
Awesome. So we have a couple of questions. Besides being viable, have you checked whether the phagocytosed oligodendrocytes are stressed or desilated?
So we looked at the PSVU dye, and that looks at negatively charged phospholipids, and they don't express that in general. Obviously, there's a little bit, there's like 10% that are stressed, but most of them are not stressed. They're not expressing phosphatidylserine or this PSVU dye, which is a little bit more general. It just labels negatively charged phospholipids. But more importantly, a cell that's stressed would probably not have a good mitochondrial membrane potential, so that DIL-C15 dye that we use probably wouldn't show up as well. But in general, they seem like they're healthy and happy, and they're being engulfed for some reason, but it's probably not because they're stressed or dying.
Great. I have a question from Longjun Wu. Any behavioral phenotype or motor dysfunction in CX3CR1 knockout mice?
Yeah, we haven't looked at behavior. It's definitely on our radar. There are other groups who have looked at behavior in CX3CR1 knockout mice in general. It gets tricky because we know that there's a difference in synaptic pruning as well. So we're really trying to focus on oligodendrocytes and not look at synapses in this model, and it's difficult to do that. I know that there are papers that have looked at behavior, but it's more in a synaptic type of realm and not in oligodendrocyte realm. So that's something that's on our radar, but we haven't done it yet. It's difficult to tease it out between synaptic engulfment and oligodendrocyte engulfment.
Have another question from Adrian Rodriguez Contreras. Have you explored the factors that determine the time to engulfment? You mentioned 20 minutes. Is this in vivo or ex vivo?
That's a good question. This was in an ex vivo slice. So I guess it could be different in an in vivo model, but it would be difficult. Technically, it's something that would be great to do to try and access the corpus callosum, but it's tough to try and get there with like a 2-photon microscopy type of thing. We haven't done it yet. It would be great to look at that, but from our data, it's suggested 20 minutes. We have seen other papers that have mentioned other cells being engulfed in a similar time frame or other material being engulfed in a similar time frame. And again, that's from the time that the microglial cell has recognized the OPC and has initiated that engulfment process. So yeah, I can't say what the timing would be in vivo, but at least comparative to the literature, it seems like it's in line.
I have another question. Might it just be weakly myelinating oligodendrocytes that are phagocytosed, just like weak synapses are eliminated?
Sure, that would make a lot of sense. We know that they're pre-myelinating, so we have to figure out what that signaling cue is next. Like what's determining if a cell is going to be engulfed or not based on other factors. So we're doing some different assays on these cells just to determine, are they expressing something that they're not going to be good myelinators, or is there a problem with too many OPCs later in life? Is that why we're getting our thinner myelin? It's a good question. It's the golden question we're after next.
And looks like one more question. Have you looked at the glycocatalyx of phagocytosed oligos?
No, we have not. That would be really interesting. I think what we need to do now is kind of profile these oligodendrocytes that are being phagocytosed. And the best way to do that is some single cell work, and we haven't gotten to it yet. But it's definitely on our radar. We have a long list. This has opened so many doors. Our lab has a lot to do.
And I just have one last question. So I was wondering what your thoughts were on why the increase in OPC survival in the 630 knockouts leads to decreased myelination. Is this sort of like, a steric space thing, or do you think there's signaling molecules involved?
Yeah, this was a huge problem with our paper when we were trying to get it reviewed and published, because we expected there'd be thicker myelin. We thought that more oligodendrocytes, there should be more myelin, and we saw less. So at first we were pretty surprised, but it kind of makes sense if you think about it. It's if we have too many oligodendrocytes, and it's kind of like a stagnant pond. It's like they're not going to all work well together. Same thing with the concept of synaptic pruning. You have, for a lot of people use the analogy of a rose bush. If you have a rose bush that has a lot of roses on it, they're all fighting for nutrients. And if you have too many of them trying to compete for the same nutrients, they'll all suffer. Whereas if you get rid of the weaker ones and have the stronger ones survive, then they'll have a better ability to survive and flourish. So probably something to do with that. That's definitely our thoughts, but we have to try and figure out what these ligands are in the signaling molecules.
Great. Thank you for your talk. That was awesome. So we can go ahead and move on to the next